Hello everyone, this is a psychiatry lecture for semester 7 medicine. Our topic today is neuroscience of psychiatric disorders. So in this lecture I hope to give you a little perspective, neuroscientific perspective for psychiatric disorders to help you better understand what psychiatric disorders are and hopefully help you appreciate more the complexity of the mind is and dispel any misconception that psychiatric disorders are not really a disease, it's just random things that can happen to people or things like that. But psychiatric disorders are actually a really serious disease, serious problem that needs proper handling. So what are psychiatric disorders? You should probably already know that psychiatric disorders are disorder of the mind which manifests as abnormal behavior. These abnormal behavior are results of significant function impairment in the brain and usually causes a cluster of specific symptoms that then we can define as a syndrome. So each syndromes usually have common symptoms that we can identify, but then the causes of this, the emergence of these symptoms can be very varied between person to person. And psychiatric disorder is a huge burden in the world today. It is the #1 contributor for disability adjusted life year, which is the number of years lived with disability in addition to the number of years lost to the disability by non-communicable disease. As you can see in this graph, neuropsychiatric disorders contribute to 28% of all disability adjusted life year. And among these, the most common psychiatric disorder is the unipolar affective disorder, followed by substance abuse, or addiction, and then there's schizophrenia, which is the most common psychotic disorder, and then also bipolar affective disorder. We've defined what is psychiatric disorder. So what is neuroscience? Neuroscience is the study of nervous system that aim to explain how behavior, perception, memory, and other functions that are generated in the brain are generated. As we know, the nervous system is a self-organizing system that is plastic. So the most common analogy is that the brain is like a computer. But this particular computer, it changes all the time, depending on our experience, depending on our knowledge, depending on what we feel, and everything that we know. So in a way, it is like a computer, but each computer are different. And this computer system, which is the brain, is responsible for all human behavior. So we can view human behavior as the output from the computer. So then if every single computer is different, then every single output cannot be the same, right? That's why people are trying to find the common theme in the brains to help us understand better how certain specific behavior are generated. So when we are studying the brain, we are studying ourselves basically. So how would neuroscience help us understand psychiatric disorder better? Well, psychiatric disorders are not any different from any other neurological disorders such as Alzheimer's, Parkinson's, multiple sclerosis, disorders that we can clearly see there are something wrong in the brain. Psychiatric disorders are the same, only just the difference, the change that are occurring in the brain are more subtle. It's harder to see, therefore we need to look deeper into the brain. But at the end of the day, psychiatric disorders are just disorder of the nervous system. It has clear neuropathological basis how something becomes a disorder. And how do we know this? There are imaging evidence such as inappropriate activation of certain parts of the brain in psychiatric patients, like schizophrenia patients shows hypofrontality in their fMRI, or inappropriate activation of amygdala in anxiety patients. And there are also volume changes in the brain, like certain regions in certain disorders usually are found to be smaller in psychiatric patients compared to normal person. And then there are genetic evidence where there are multiple interactions of genes that, along with the effect from environment, can create a disorder. For example, PTSD. 30% of risk for PTSD is determined by inheritable factors, which is the gene. From a genome-wide association study, there are hundreds of genes involved in schizophrenia. So there are genetic evidence that shows that some genes makes people more susceptible to some psychiatric disorder. And then there is neuroplasticity, where we know that a mistake in functional reorganization of the brain can lead to the psychiatric disorders. And there are evidence that people that are experiencing psychiatric disorders have decreased neuroplasticity, such as in depression, OCD, and others. There's a decrease in cognitive flexibility. And neuroplasticity is also why therapy and drugs work. It's the organizing of the brain to restore it to the correct organization can then fix the disorder. Then how does neuroplasticity can lead to a disordered mind? It is a culmination of the biopsychosocial interaction. Like in this slide here, on the right we have the biological component, neurotransmitter, protein, hormones, cytokines, and genes. On the left we have the environmental components, physical environment, nutrition, caring pattern, information, and lifestyle. And then all of these interacts to form who we are, to form our brain, our mind. And as you probably know, everything needs to exist in balance. So when a balance of some of these stuff are disturbed, maybe some people lack neurotransmitter, lack certain neurotransmitter in their brain, or they're experiencing trauma, extreme stress, or other stuff like that. And then the organization of the brain can be disturbed and create a disorder. So we know that our brain's neuroplasticity, depending on the state our brain is in, and the environment, etc., can lead to disorder. And the most obvious instance of neuroplasticity happens during learning, which is the process where our experience and the things that we practice cause direct change to our nervous system, which then leads to behavioral change. And when these changes are retained, when the experience are retained, it forms memory. Therefore, memory is the basis of what we are. Louis Buñuel says that you have to begin to lose your memory, if only in bits and pieces, to realize that memory is what makes our lives. Life without memory is no life at all. Our memory is our coherence, our reason, our feeling, even our action. And without it, we are nothing. So basically, at the core, memory is everything to us. How we perceive things, how we process things, how we react to things. It all depends on what we learn and what we form as memory. And learning and memory formation happens in all animals, from reptilians to mammals to human. Although they can have different motivation for learning. Reptilian brain usually only learn for security purpose, so they are only reacting to their environment, how to survive. Mammalian brain, which is more advanced, can have motivation such as protecting their children or um, preserving their species. But the human brains is even more advanced. We have creative thinking. We do stuff that can be in contrary to our immediate interests, but then can be beneficial for us in the long run. But although learning and memory evolves from more simple organism to us, that has the most complex brain of all animals, the mechanism are largely retained. So learning mechanism from reptilian brain to human brain are basically the same. So since learning mechanism is conserved from lower organisms to humans, it is useful to learn how the mechanism is in simpler organism, and the findings can still be applicable to the mechanisms in human. And one of the earliest clue to learning mechanism was from the study by Eric Kandel and James Schwartz in 1960s and 70s. In their experiment, they studied this sea slug called Aplysia californica, and it's a relatively simple organism with large neurons that they can easily observe, and it has a unique reflex, which is the gill withdrawal reflex, where normally this animal has its gill outside and exposed to the environment, as shown in the B picture here. But when either its head, tail, or the mantle or siphon is touched by anything which elicits a stimulus, then it has a reflex that will retract this gill into its body so that it's protected from the environment. So the learning that they observed in the sea slug was concerning this with gill withdrawal reflex. And what they found was the extent of the reflex, like how strong the reflex is shown, can be varied. And they call this habituation and sensitization. Habituation is when the reflex gets weaker and weaker the more you stimulate the animal. So if the mantle is repeatedly touched within a short period of time, the reflex that is shown by this animal gets weaker and weaker, as shown in this graph above. And if you keep stimulating this animal again and again over a long without giving it a certain amount of rest, then the habituation is retained. It can even be retained for a couple of days or weeks. But if you stop stimulating it for a while, for say giving it a rest for 2 hours, the normal response can be returned. So that's habituation, where after multiple exposure to a certain stimuli, the extent of the reflex, how strong the reflex that is shown gets weaker and weaker because the animal learns that the stimulus that was given actually doesn't do anything. It's not an immediate danger to itself, so there's no need to um, do the reflex. Then on the other side of this habituation, there is sensitization, where after given a first stimulus, and after given a first stimulus and the animal give a reflex response, um, the researchers give another stimulus, but in a different area of the animal's body, so that it sensitizes the animal for receiving some kinds of stimulus, which in this case is a strong stimulus to the tail, and this sensitizes the animal to touch. And when another stimulus is given to the mental, the response, the reflex response that was shown by the animal is actually stronger than the first one. So in the case of sensitization, the Aplysia californica learned that there's other stimulus in the environment, in its immediate environment, that can possibly be dangerous to its gill, such as the strong stimulus to its tail. So when the gill is then stimulated again, the animal learns that the animals assume that the second stimulus might be similar to the stimulus it's received on its tail, and it felt that the stimulus to its tail was quite strong and possibly dangerous, so that the withdrawal reflex that it shows is stronger than the first reflex, and that is called sensitization. So this is the circuit that is involved in the gill withdrawal reflex in aplysia. For habituation, it only involves the sensory neurons from the mantle and siphon, and synapse with the motor neuron that innervates the gill, which then elicits the withdrawal reflex itself. So in habituation, the sensory neurons actually become less and less sensitive to the stimulus that was given, so then there's less response in the motor neurons, which means then a weaker reflex response in the gill withdrawal. While sensitization, on the other hand, is a little more complex, since it involves sensory neurons from other parts of the animal, such as the tail or the head, and those sensory neurons then synapse with an interneurons, which then synapse to the sensory neurons, the sensory neurons that was from the mantle and siphon, and it can then alter the transmitter release from these presynaptic neurons. And the mechanism that they found was that it involved release of serotonin from the interneuron to the presynaptic neuron, and the serotonin activates the G-coupled receptors, which then activates adrenal cyclase that turns ATP into cyclic AMP, which then acts on protein kinase A, which in turn phosphorylate the potassium channel, and it basically closes the channel and then prevents potassium from leaking out of the cell. This means that it's prolonged depolarization and allows calcium to enter the cell more. And since there are more calcium, then the cell can release more neurotransmitter, which then in turn activates more receptors in the postsynaptic motor neurons, and it can elicit more stronger reflex response. But as we saw before, This mechanism only accounts for a short-term alteration in response. Like previously shown, after not being stimulated for a couple of hours, then the strength of the reflex that is shown by the sea slug can return to normal. For longer term change, synapse alteration must occur. So there must be a change in the structure of the synapse. It can be a change in the number of receptors, or it can be a change in the amount of neurotransmitter that is um, released. The longer term synaptic change is called long-term potentiation for changes that leads to stronger response in the postsynaptic neurons, or long-term depression and changes that leads to weaker response in the postsynaptic neurons. And the mechanism of the two are similar, only with different end results. But both most certainly involve signaling from the postsynaptic neurons back to the presynaptic neurons. And here we'll focus only on the long-term potentiation. So long-term potentiation involves two channels, non-NMDA channel and NMDA channel. These are both ion channels. The non-NMDA channels usually only allows sodium to come in, while the NMDA channel allows both calcium and sodium to come in. For the NMDA channel to activate, it needs sodium to come in first from the non-NMDA channel, and it allows the removal of this magnesium block that is usually present in NMDA channels. And when this magnesium block is removed, it then allows calcium to come in. Calcium will then react with a protein called calmodulin, which then activates several kinases such as the calmodulin kinase, the tyrosine kinase, and protein kinase C. And then calmodulin kinase can potentiate the non-NMDA channel by phosphorylating it and allowing more sodium to come in. And then the other one is PKC, protein kinase C. It also can potentiate the non-NMDA channel, but PKC can also lead to the production of nitrous oxide, which then becomes a signaling molecule to signal to the presynaptic terminal to increase the transmitter release. So there are more transmitter that are released, there are more channels that are activated, and the activated channels are also phosphorylated to allow more sodium to come in. Therefore, it can lead to stronger postsynaptic response. For long-term potentiation, or LTP, to last longer, it requires the formation of new synapse, which also involve gene expression, as can be seen in this diagram here. It also involves the calcium from NMDA channels reacting to activating calmodulin, but in this case calmodulin then activates the adenylyl cyclase, which can produce cyclic AMP. Cyclic AMP can then activate protein kinase A, which in turn can activate SCRAP, which is a transcription factor. It's short for cyclic AMP response binding protein, and CRAP then allow the DNA transcription to mRNA, which then synthesize protein and can form new synapse and new channel for the neurons. So that was the basic of how learning occurs in animals. How then learning happens in mammals? What we know so far is that it depends on the hippocampus. We know that the hippocampus is important for memory formation in mammals, especially human. Um, from the case of this famous patient called patient HM, which has been extensively studied. This HM person had bilateral removal of his hippocampus. It was done to help his condition, which was that he was having severe seizures, and his doctors thought removing his medial temporal lobe can help reduce his seizures. However, after his surgery to remove his hippocampus, HM then can't form new declarative memories. He still have all his previous memories, he still remember them, but he can't make new memories, especially declarative memories, which is memories about facts, like for example what you are learning today in this lecture. He can't remember that, but he can make procedural memories such as how to ride a bike, for example. So the fact that memory formation is impaired in patient HM clearly shows that hippocampus is central to the formation of memories, especially declarative memories in human. This is an MRI of patient HM's brain with compared to a normal person's brain, which was age matched with patient HM. It shows here that clearly in the medial temporal lobe, the hippocampus in HM is removed, while in the control patient, the medial temporal lobe is intact. And here it goes further back from the brain. It shows that the medial temporal lobe, or especially hippocampus, is still missing in HM. And as we go further back, we can then see that the structure starts to look more similar to from HM and the control, showing that only the medial temporal lobe, especially the hippocampus head and body of HM, was removed. The mechanism of learning itself in the hippocampus is still basically the same. It's still LTP, long-term potentiation and long-term depression. It involves the glutamatergic transmission and the NMDA channel. And long-term potentiation here can be associative, which means it requires it involves more than one stimulus. So it's involve coupling of stimulus into one potentiation, or non-associative, which is just one kind of stimulus being given repeatedly. And here, both the presynaptic and postsynaptic neurons must be active for long-term potentiation to happen. And this is an example of an associative potentiation. It's basically similar to what happens with the aphasia before with sensitization. When the circuit is given a potentiating stimulus, then the response that can be recorded after the potentiating stimulus will be stronger compared to the response that is given when the circuit is not given any potentiating stimulus. And here we can see the voltage from the postsynaptic neuron control compared to the one that is given the potentiating stimulus. It shows stronger response, higher current, and the basic principle, the mechanism, is still the same with LTP before, which involve either phosphorylation of channels, making it more sensitive, allowing more sodium in, or formation of new channel and new synapse. So how do we know that long-term potentiation, or LTP and LTD, is the same with the formation of memory? It is shown in this experiment using mouse and something called Morris water maze. In this experiment, a mouse is put into this container which is filled with water. Usually before the experiment, the mouse is shown that there is a standing platform somewhere in either one of these quadrants, so that the mice can avoid being in the water. So after being shown where the platform was, usually the platform is removed and then the mice is put back in the water maze. And usually when the mice remembers where the platform is, it will go look for the platform in the area where the platform was before, which is what happens in normal mice in the control here. But when NMDA receptors is blocked, so we are blocking long-term potentiation in the second mice here, this mouse just doesn't look for the platform in any particular place. It just goes around everywhere in the water maze, which shows that it has no memory of where the platform was before. Therefore, in this case, blocking LTP equals to blocking memory. So far we've learned how memory itself is formed, and in the case of the mouse in the Morris water maze, how memory can lead to behavior. But we also know that memories and behavior can be strengthened or weakened depending on the consequences of said behavior, as well as the, for example, emotional value of the memory itself. And one of the way the degree of a behavior is shown can be altered is by reinforcement. This is where favorable consequences from behavior can lead to the organism learning that such behavior have favorable consequences, so that the behavior is exhibited again and again. And this involves the release of dopamine from nucleus accumbens in the basal region of the forebrain. And this is also the mechanism that can lead to addiction in the case of consumption of amphetamine, cocaine, opiates, marijuana, caffeine, and nicotine that promotes the release of dopamine. So in the case of addiction, the person learns that consumption of these substances can lead to a pleasant feeling. So the behavior is repeated again and again. This is one example of such behavior, of addictive behavior. In this experiment, the mouse is implanted with an electrical rod that stimulates directly in the lateral hypothalamus to induce the release of dopamine in its brain by pressing this lever here. And since the animal learns that pressing this lever leads to this pleasant stimulation of dopamine release, the mouse in this experiment repeat the behavior of pressing this lever again and again and again, and even ignoring its need to eat food and drink water. Then when a dopamine antagonist is introduced into the mouse, so the stimulation doesn't lead to release of dopamine anymore and doesn't have any pleasant effect for the mouse. This behavior of continuously pressing the lever stops. So this is an example of um, how an animal can learn that some behavior can lead to uh, pleasant consequences or favorable consequences, so that it learns to reinforce such behavior and repeatedly do this behavior again and again. So since our brain continuously receives stimulation, continuously learn, continuously um, form and reinforce certain behavior, and continuously adapt to whatever experience we have, it is no wonder then um, everything that we've experienced in the past can um, affect us in the future, such as this finding where they find that childhood adversity can lead to increased risk in adulthood disease. Here they examine children that experience adversity during childhood, such as poverty or abuse during their childhood, and follow them until their adulthood, and find that children that experience these difficulties during childhood are more likely to experience disease in adulthood, including major depression and cardiovascular risk and metabolic risk, or a combination of these various disease. So now let's move on to specific psychiatric disorder that we know involves learning. First, anxiety disorder and PTSD. So anxiety and PTSD involves fear, which in normal condition is a behavior that we need in order to deal with perceived threat. This behavior helps us survive in the environment and keeps us away from danger. However, in disordered condition, the fear network can be activated inappropriately. And we also note that this network can be conditioned, which means it involves learning. And learning in the fear network is mostly associative learning, which is also similar to the example of classical conditioning in Pavlov's dog's experiment. So in Pavlov's experiment, he make a dog associate one stimulus with another, which then with an unrelated stimulus that can then elicit a conditioned response. In this case, unconditional stimulus of presenting the dog with food give the unconditioned response of salivating. But then when he presented the unconditioned stimulus of food along with the conditional stimulus of ringing a bell, eventually the dog learns to associate the bell ringing and the food will come together, and eventually the dogs will shows a conditional response where it responds to ringing of the bell with salivation, which becomes the conditioned response. Associative learning is what happens in the fear network centered around the amygdala, which is the emotional center of the brain. Similar to the example of Pavlov's dog in classical conditioning before, fear network can also associate certain sensory stimulus with an unconditioned stimulus that are negative, such as, for example, coupling the sound of this will create a conditioned response where eventually when the animal hears the sounds of a bell, it will exhibit fear or anxiety of being shocked by an electrical stimulus to the feet. And we know that the amygdala is central for this response, because when the amygdala is removed from an experimental animal, for example, this fear conditioning is also removed. So there's this fear conditioning behavior is abolished. And here we see that the amygdala has extensive connection to the hippocampus, which means the emotion that is associated in the amygdala can be consolidated into memory via the hippocampus. And then we also see that there is a short route to activation of the amygdala, and there is a long route, which is through the medial prefrontal cortex. So this is why sometimes the emotion of fear can be activated pretty quickly, but then as we think about what the stimulus actually is, which involves the medial prefrontal cortex retrieval of memory and logic, sometimes we think that such fear is actually not necessary. It's therefore can be suppressed by input from the medial prefrontal cortex. And then we also see that the amygdala has connection to the lateral nucleus and the HPA axis, the hypothalamus, pituitary, and adrenal axis, which these are the output from the fear response from the amygdala, which then stimulate the autonomic nervous system, so the flight or fight response, and also the stress response, cortisol production. And this diagram shows that the learning process itself of the coupling of the stimulus as fear, if itself happens in the amygdala, from the lateral amygdala to the basolateral amygdala, and then activation of the central amygdala leads to the expression of the fear. So connection from central amygdala to lateral hypothalamus leads to increased heart rate and blood pressure, and then connection to the dorsal vagal nerves leads to bradycardia or ulcers, and then connection to parabrachial nerves leads to panting, respiratory distress. Connection to basal forebrain leads to arousal, vigilance, and attention. Connection to reticular pontis caudalis leads to increased startle response. And then central gray area leads to freezing, social interaction. And paraventricular nucleus leads to corticosteroid release. So those are the output of what we learned as fear in the amygdala. And what leads to anxiety and PTSD is inappropriate activation of these connections to stimulus that are not supposed to activate them. And since the learning itself occurs in the amygdala, especially in the lateral amygdala, decoupling of this associated fear and stimulus also needs alteration in the network in the lateral amygdala. So with the advance of imaging technique and volumetric technique to measure volumes of parts of the brain, we've been able to more conclusively implicate the amygdala in anxiety disorder. One example is this paper, which found a reduction in amygdala volume in children with anxiety disorders. Specifically, they found that the gray matter in the left amygdala has lower volume in children with anxiety compared to the control subjects who obviously don't have anxiety problems. And this is just a picture of the result of the technique they use, voxel-based morphometry, where they compare the basically the average volumes of each region of the brain from one group and a control, and they found that the left amygdala, specifically in the anxiety group, was found to be significantly smaller compared to the control group. And this is also a similar study, but in adults, adults with panic disorder. They also compared the brain with healthy patients, healthy comparisons, and found that amygdala, both right and left amygdala, were smaller in patients with panic disorder. And they also find a negative correlation between anxiety and amygdala volume. So the worse the anxiety in these patients, the smaller the amygdala volumes that they found. So by showing that in people that have panic disorder and anxiety disorder, the amygdala volume is altered, it proves that there is a connection between these disorders and amygdala, although it doesn't really, we still don't know yet whether the decrease in volume is the result of anxiety and panic disorder, or is it the cause for anxiety and panic disorder. And this picture is similar to the previous one. This is the result of the voxel-based morphometry, where they specifically find a change in volume in the right amygdala of the patients with panic disorder.
And as previously mentioned, the hippocampus is also part of the fear network. Hippocampus have extensive connection to the amygdala, and its position anatomically is very close to the amygdala, so it is no wonder that it plays an important role in the fear network. So the hippocampus is important for contextual memory, which then in turn have a big role in contextual fear. And we also know that the hippocampus is affected by stress such as anxiety. More specifically, the CA2 region of the hippocampus shows dendritic loss. So the neurons there shows less dendritic spines and less branching. This in turn would obviously has an effect to learning. And context here, in terms of contextual fear, it's actually very similar to the condition response, similar to Pavlov's dogs before. But in this case, instead of a specific stimulus, which was the condition stimulus, the thing that triggers the condition response is actually contextual, such as maybe the room that the animal is in, or a certain action that it has to do, or presentation of a certain thing like temperature along with smell, or any combination of stimulus. And once this context is learned and coupled with the thing that causes fear, it becomes contextual fear, and the response can be exhibited without the presentation of the actual stimulus before. And since fear is actually a learning process, fear is learned, uh, to erase that fear, in other words, fear extinction, is also an active learning process. So fear will not disappear on its own, but we need to learn for ourselves that what we are fearing is actually not dangerous for us. Therefore, we don't need to fear it. And therefore, the learning process and the hippocampus is also central to this process of fear extinction. And this is also why exposure therapy for phobia and any fear-related disorders actually works. So when we present the patient with the conditioned stimulus or whatever it is that they're fearing, without the negative consequences of what they're fearing, which is the unconditioned stimulus, then the patient can start to learn to uncouple the coupling from the conditioned stimulus and the unconditioned stimulus and make new connection, that the conditioned stimulus is actually something that is something that is not to be feared. And this experiment on the right actually shows that acquiring fear and forgetting fear is a learned process. It's a learning process. So in the first experiment, um, this is in mice again, and the experiment was similar to classical conditioning. So the first one couples a tone or a sound with an electric shock. So the conditioned stimulus is the tone, unconditioned stimulus is the shock, and once the animal learn to couple tone and shock together, when only the tone is presented, then it'll show the conditioned response of freezing, similar to when it is when the animal is shocked. So here, to block that learning process, the experimental administer a protein synthesis blocker called anisomycin. And so in the vehicle animals that wasn't given any anisomycin, when the tone was given a second time, the response was still quite similar to the first response. But when anisomycin was administered, after the second test, after the anisomycin was administered, the reaction was a lot less compared to the first reaction. And therefore, this shows that the retention of memory is blocked by blocking the process of protein synthesis in the amygdala, which is likely required for either for potentiating the channel proteins or creation of new branch and new channel proteins. While the second experiment here shows the role of hippocampus in contextual fear learning. So the experiment is the same. It couples a contextual situation with a shock, and then present the animal with context only after 24 hours, and then either administer anisomycin or vehicles into the animal's hippocampus, and then present the context again as the second test. Here what they measured was the latency, or how long the animal realized that the context is the same with the context that they learned to be coupled with the shock. So in the vehicle animal, the latency is relatively similar or almost the same between test 1 and test 2, while the animal that was injected with anisomycin shows much longer latency to present the response in the second test after the administration compared to the first test. So these two experiments clearly shows that there is some sort of protein synthesis going on, which is required for learning, and either neuronal branching or creation of new protein channels, as well as potentiation of channels. And since fear extinction is an active learning process, the mechanism is similar to any learning process before. So it is facilitated by an NMDA receptor, and it is proved by experiments that shows that infusion of NMDA receptor antagonist to the amygdala can block both acquisition of fear, so learning that something needs to be feared in the first place, as well as extinction of fear, or trying to learn that the stimulus is something that is not necessarily something that is to be feared. And secondly, when an agonist is infused to the amygdala, it can also help facilitate learning extinction, fear extinction, when coupled with training. So administering agonist helps with learning, so it can help the process of fear extinction by promoting neuroplasticity and interfering with the reconsolidation of the fear that was already learned previously. And now let's move on to the second psychiatric disorder that is closely related to the learning process: major depressive disorder. Major depressive disorder is a mood disorder, which typical symptoms includes feeling of sadness, feeling worthless, and loss of interest in hobbies and stuff, change in appetite, and some other stuff, including suicidal ideation, tiredness or sleep disturbance, and etc. And the causal factor for major depressive disorder, or MDD, are genetic and early life adversity. Early life adversity is especially important here, since this shows that what our brain experience in the early life can shape us as a person. And since every person's experience are different, and there are no two brains that are the same, so our brain exists in a spectrum based on our genetic makeup and our experiences, and these two can determine how resilient we are to stress. So in other words, how susceptible we are to depressive disorder. And MDD is a system level disorder. It involves multiple neurotransmitter system, including the serotonergic system, which is usually hypofunctioning. And this is thought to be related to dysfunctional attitude toward oneself and toward the world in MDD patients. And the evidence that there is a serotonergic hypofunction in MDD is that in MDD patients, it was found that the brain has low metabolite level for serotonin, and also there's an upregulation of receptor expression. So there are more serotonergic receptor in the brain of MDD patients as compensating mechanism due to the low level of serotonin that is available to them. And then the second system is the noradrenergic system, and there is mixed evidence here. There is evidence that they are hypofunctioning, but there is also some evidence of hyperfunction of the noradrenergic system. And the evidence for the hypofunction is that there are fewer neuroadrenergic neurons in the locus coeruleus of uh, MDD patients that committed suicide, and then there's also lower level of the metabolites for noradrenaline, which predicts suicidal behavior. However, in patients that actually committed suicide, it is also found that the alpha adrenergic receptor binding is higher in these patients, so which suggests a hyperfunction of the receptor. And lastly, the third system is the dopaminergic system, which is known to be hypofunctioning. And this impaired the reward system and promote anhedonia. So the loss of interest symptoms is primarily due to the dopaminergic system malfunction. And MDD also affects what region of the brain, including the prefrontal cortex, the anterior cingulate cortex, as well as the limbic system. And there are evidence that MDD disturb memory formation. And this is one such experiment that demonstrates that MDD can disturb memory. So in this experiment, there are two groups of mice, the control, and then mice that are treated to CUMS, which is chronic unpredictable mild stress. This CUMS is known to simulate stressful life condition, which if treated in a prolonged amount of time can cause the mice to exhibit signs of depression. And the memory that is tested in this experiment is the spatial working memory. It was tested using the Morris water maze that was already explained before. So this test measured the amount of time the mouse was uh, uh, the amount of time the mouse spent in the correct quadrant in the water maze. So the higher the number, the better it is. And here we can see before the treatment, all mice perform similarly to each other. There is no significant difference between the time they spend in the correct quadrant in the water maze. But after treatment for 21 days, it significantly dropped compared to the control. The same with the after 41 days, it's also significantly dropped compared to control. Although in mice that are treated with CUMS from 21 days to 31 days to 41 days, the amount of time actually increased, which might reflect some sort of habituation or adaptation behavior in the mice. But overall, this shows that the working memory is indeed impaired in the depressed mouse, and compared to the control mice, the performance obviously decreased. Another way MDD affects learning in depressed subjects is by impairing cognitive flexibility, which is the ability of an animal or someone to change strategy to deal with an unexpected condition by updating their knowledge, by incorporating new information into what they already know. And this can be measured in mice with an experiment, which is the attention shifting task experiment, where mice are tasked with finding food which is associated with a different smell or any other media. So the mice will need to learn to couple the information of where the food is with the new information of smell or any other media that are actually unrelated to the smell or food. its food themselves. And here we found that cognitively impaired mice, or mice that is treated with comes to simulate depression, actually requires a higher number of trial and error. So they can't um, make the new connection between uh, the new information, which is either the smell of or any other media, with the placement of food that they need to find. And the experiment is the same: animals with control groups and animals treated with chronic unpredictable mild stress. And before the treatment, they're all basically the same, but after treatment with comes, after 21 days, the number of trials increased significantly compared to the control, and uh, after 31 days also higher than the baseline before, and then after 41 days also still significantly higher number of trials compared to controls, although again there is a decrease in number of trials, so there's an improved performance from 21 days to 41 days, which suggests that the animals was starting to get used to the CAMS treatment, so it becomes less stress, it becomes more capable of performing the task better. But this still shows that during depressed condition, um, cognitive flexibility is clearly impaired. So how do these impairment then come to be? It might have something to do with cell plasticity and cell survival. So we know that in MDD patients there is serotonin hypofunction, and then it was also found that serotonin acts as a trophic factor via its interaction with G protein coupled receptors, which promotes cell survival. Therefore, when there is less serotonin, there is less trophic factor, so there is less signals that tells the cell that they need to keep surviving. And as shown with the experiment with the mice before, cognitive flexibility and working memory is impaired in MDD patients. This is because those functions are mediated by the hippocampus, and the hippofunction in MDD affects hippocampus greatly because serotonin receptors are highly expressed in hippocampus, so there is an extensive network of serotonergic neurons in the hippocampus. And there are several receptors that are important for cell survival, neuronal survival. They are highly expressed in the hippocampus. First, the serotonin receptor 1A, 5-HTR1A. This receptor activates the AKT-NPI3K kinases, which regulates angiogenesis, growth conformation, and cell proliferation and survival. And activation of these receptors also inhibit GSK3 beta, which when expressed stimulates the caspase 3, which then triggers apoptosis in cells. And then there is the serotonin receptors 2A, which stimulates DNA synthesis, activating kinases ERK1 and 2, which usually results in proliferations of neurons. ERK is known to have a role in learning and memory, especially formation of synaptic and neuronal plasticity. So ERK is one of the factors that allows the formation of new synapses, and therefore is important to learning. So when this receptor is hypofunctioning, uh, the learning process can also be impaired. And then there's also the serotonin receptor 4, which activates CRAP,
which I already showed previously. Crop is important in formation of um, new branch of dendrites, as well as expression of new protein channel receptors. And crop can also increase neurogenesis as well as survival of neurons. And polymorphism of any of these receptors is associated with the MDD condition, and in patients that commit suicide, expression of these receptors are found to be altered. So therefore, the impaired serotonin signaling doesn't only impair the signaling process itself, but it also affects cell survival and therefore can affect the volume of the brain region that is affected. And this is a comparative study that showed proof that MDD condition actually cause an alteration in brain structure or brain volume. And this study compared MDD subjects against healthy control, and they compared the gray matter volume of each of these subjects. They compared in the MRI scans of the brains of the subjects using voxel-based morphometry, which is a method to compare brain volumes from MRI. So what they found was in the MDD group, although as a whole there is no significant difference in brain volume between MDD and the healthy controls. In some specific regions, they did find significant difference, such as in the pre-supplementary motor area, which was found to be smaller in MDD patients. And they also found correlation between hippocampus volume, both the right and left hippocampus. It was positively correlated with symptom severity, which means that the hippocampus volume is related to the characteristic of the depressive symptoms that is exhibited by the patients. And they also found that the longer the patients have MDD, the lower the volume of their hippocampus was, which shows that MDD condition can progressively cause neuronal cell death in the hippocampus, which eventually leads to the volume reduction. And since the MDD patients were first episode MDD, so they were experiencing depression for the first time, and they were drug naive, so they haven't been medicated yet. This shows that it was actually the depressive condition itself that is leading to this alteration in brain volumes and not the medication. And these are just the pictures of the results of the paper. A is the significantly smaller pre-supplementary motor area in the cortex of MDD patients. And then B is the correlation results, where the hippocampus, both right and left, are correlated to the severity score of the MDD patients. And if the previous study showed structural correlate to MDD, this one shows the function correlate for MDD, which is shown by imaging using the resting state functional MRI, specifically regarding the connectivity in the default mode network. So what they are looking at is the connectivity in this network when the subject is actually not performing any specific task
which is why it is called the resting state, because the patient is in a rest state. And they are looking at two things that are commonly found in depressive patients, which are rumination and overgeneral autobiographical memory. So rumination is obsessively thinking about something repeatedly, so thinking about something, overanalyzing something again and again and again. While overgeneral autobiographical memory is the phenomena where MDD patients usually unable to retrieve specific memory when asked about something, so they overgeneralize their memory. So in this study, they found an increased functional connectivity in the anterior medial cortex region, specifically the medial prefrontal cortex and anterior cingulate cortex, which they correlate to the rumination symptom. While on the other hand, they also found a decreased functional connectivity in the posterior medial cortex regions, specifically the posterior cingulate cortex and precuneus, which they correlate to the overgeneral autobiographical memory. And these are the result pictures. These two are comparing the connectivity of the default mode network. And here it clearly shows increased connectivity at the anterior medial cortex region in the depressed subjects compared to the controls. While the picture in C shows the difference between depressed and control, which was negative at these regions. So this increased connectivity was correlated to the rumination in depressed patients, while this one's the decrease in connectivity was related to the overgeneralized autobiographical memory in depressed patients. So in conclusion, today we've learned that learning and neuroplasticity is something that occurs normally and it is essential for normal function. But we've also learned that when these processes are disturbed or impaired, it can lead to disorders such as anxiety in case of impairment in learning, making the wrong association between stimulus and such. And also we also see that in depression there is an impairment in learning as well, which is shown by the mice in the Morris-Water maze, also in the attention shifting task. And if we know where the impairments are, it might be possible for us to restore this function. So if we can then try to restore these abnormal functions, we may then restore the brain to the normal state, and so it can function normally again. So in a sense, curing the psychiatric disorder. So to do that, we need to understand these mechanisms better. So we need to isolate where exactly in the brain is there anything going wrong. Is it molecular? Is it in level of cellular? Is it already in the system level, or is it everything together? So we need to figure out which comes first, and if we can then create a target that are specific for a certain condition, we can then create a better therapy for such conditions. Because the current therapies, such as antidepressant therapy, for example, are generally found to be not specific enough. So there are a lot of side effects, and there are sometimes medication that doesn't actually work, because obviously depressions are not all the same. So if we can understand everything better and figure out the specific mechanisms that we can target, we can then make better therapy for the patients. And that's it. Thank you.